This is IAL Biology, and today we're working on one of the core practicals studying membrane permeability. We're using beetroots for this experiment because the cells of this plant contain a pigment can be easily observed. We will start by cutting a beetroot and making cylinders using a cork borer. Following this, we will make sure that these cylinders have equal length, so that would be a fair experiment. Following this, we will rinse those cylinders just to make sure that there is no pigment remaining on the surface. Right after this, we will soak those cylinders in water bath for about 20 minutes. Following this, we will transfer the fluid and determine the degree of color. We will now use a colorimeter to determine the color intensity. The higher the absorption of light in the colorimeter, the greater the amount of pigment and the higher the membrane permeability. To summarize the process, we will first collect the cylinders from one beetroot. Then we will cut them all equally to make sure it's a fair experiment. Following this, we will rinse those cylinders and incubate at different temperatures. Right after this, we will transfer the fluid into the colorimeter and we will determine the light absorption. The greater the absorption of light, the higher the amount of pigment.